Hi everyone, this is Dr. Rich at the College at Brockport. I'm going to do a short recap of the blood vessel lab. I want you to be ready for this next lab exam. This is a really strong combination of anatomy and physiology. You have to know them both, and if you ignore what I wrote or if you ignore the textbook, you're going to get hurt really bad. You need to know how to identify things, but also how they work. We've had some exercises in lab, and I really hope that you do well on this next lab exam, and after spring break that you do well on the lecture exam. Cardiodynamics and this dynamic control of blood flow is a challenge. Let's get right to it, because I'd like to try to keep this short. So I always tell you, look at the objectives. I have that up on my screen, and it's on your screen. So objectives. Identify major histological features of arteries, capillaries, and veins. So you were supposed to look at this in lab. Maybe you didn't. The place to start now is the textbook. So take a look at blood vessel anatomy. I grabbed this slide from the lecture notes. You've got arteries, veins, and capillaries. You know arteries carry blood away from the heart, veins return it to the heart, and capillaries are called exchange vessels. Let's look at the capillary. Oh, it's not even here. But what we know is they're very, very thin, just one layer of endothelial cells connected by this, I call it a tongue and groove joint. If you ever did any woodworking, you know there's a tongue and it fits into a groove on the next piece of wood or on the floor material. Same thing for an endothelial cell, and it makes them tough. They're hard to pull apart. But what about arteries and veins? What I know from the lab is I've got a list of terms right on page 1, and included is the lumen. That's the inside. There's the tunica intima, the tunica media, and the tunica externa. Tunica means coat, so there's the external coat, the internal coat, and of course the middle layer. These are layered in these slides. You can look at the relative proportions, and you can very easily see that a major difference is that we have elastic fibers in arteries. We do not have them in veins. We've got this really thick layer of smooth muscle in arteries. In the corresponding vein, it's much thinner. Most veins on cross-section or transverse section will be collapsed. You can certainly see that on this picture from the textbook. So I'm going to go to the next slide on my slides, and what we can see is an image that I posted on Lab Images and that was on the iPad. This is of the human vena cava, and we can see the different layers of tissue, so interna, externa, and media. We can see the connective tissue layers. We can see the muscular layers, and you know this layer just next to the lumen where my mouse is the endothelial cells. The other place to look for some good images is online. Now, all of these pictures online, they've been colored. They've been digitally remixed to make the pinks pinker and the blues bluer. If you're not a histologist, I don't expect you are, the dark blue, that's the DNA, which is located in the nucleus. So it's a very small organelle somewhere inside of the cell. The pink color, or reddish color, is inside of a cell, and those are proteins in the cytoplasm. We know that red blood cells look red with this H&E staining, hematoxylin and eosin. That's because they're stuffed full of hemoglobin. We know muscle cells are quite red. That's because those muscle cells have myofibrils. Now let's look at these arteries and veins. Clearly the vein has a bigger lumen, a bigger inside compared to the corresponding artery. And you will learn that arterioles and venules, those are roughly the same part of the vascular tree. The venules have a larger diameter. Compare the aorta and the vena cava. The vena cava has a larger diameter. You'll learn why pretty soon. Look at the different layers. In this adventitia, that's connective tissue. We know this is a nerve and not a blood vessel because there's no lumen. That makes it pretty easy. Okay, I want to move on. Here's another slide. We can see in fat tissue, we have a small artery, a small vein, and really teeny capillaries. Inside of the artery, we can see red blood cells in this particular slide. In this one to the left, we don't see any red blood cells in this capillary. Of course, the walls are quite thin because a capillary is an exchange vessel, and diffusion is only good over short distances. Down here in the lower left panel, we have a transverse section, a sagittal section, cut the long way of a capillary, and we can see the stacked up red blood cells. Okay, and we can also see it's about 8 microns in diameter, correct? Let's move on. Next objective, identify major systemic arteries, veins, and the organs they perfuse using the models. I will ask you about these blood flow pathways using models, using pictures from the textbook that are not labeled, or even using a flow chart. So you have to imagine you're in a blood vessel and you're a red blood cell. This is how I do it. It's your business how you do it. But I like to sort of weave my way through. So imagine you begin in the left ventricle. We're going to leave that left ventricle via the aortic semilunar valve, and we will enter, let's move to the slide now, the ascending aorta. If we're going to go to the right arm, we must enter the brachiocephalic trunk, the right subclavian, ultimately the axillary, maybe the brachial. I think this is the brachial, and then the brachial splits. I'm not interested in all of these blood vessels. You do need to know probably the radial and the ulnar blood vessels. Just because the radial is near the thumb side, the ulnar is the other side. It's more medial. Now, how does that blood get back to the heart? So you're going to have to flip through the pages in the book just like you did in lab. And what you learn is there are corresponding veins. So there's an ulnar vein and a radial vein. Of course, the radial is more lateral. There's the median cuboidal vein where we sample blood usually. You definitely need to know that one. Then we've got the brachial and the axial, right? Here's the axial. There's the brachial. And these guys converge on the subclavian. The brachiocephalic enter the superior vena cava, which drains into the right atrium. Okay, we've just gone through the systemic circuit. We began in the left ventricle, and we ended in the right atrium. We know that the arteries carry blood away from the heart, veins return blood to the heart. That's by definition. We also know that in the systemic side, arteries are oxygenated and veins are deoxygenated, and it's just the opposite in the pulmonary circuit. When you take your exam, tell me that this is the right arm. I need to know that you know, so please put that down. I'll remind you, but you must put it down to get credit. You also need to always tell me if it's a vein or an artery. You know, some students see blue and they think vein, but you know that's false because veins in the pulmonary circuit are red. They're oxygenated. They carry blood carrying oxygen. So I always need to know artery or vein and left or right. Okay, let's move on. I'm sorry if this is too fast. I'm just trying to hurry because it's Sunday and I'd like to get home. We've already gone through 8 minutes. Back to the objectives. Here's a hard part, I think, for most everyone. Understand that blood flow is proportional to pressure and inversely proportional to resistance. So the first lesson is really simple. I think we all get it. We know that flow goes up when pressure goes up. That means if we need to increase flow, flow can be thought of as cardiac output. The heart can generate more pressure. Simple. We also know that if we need to get more blood to a region, we don't need to change pressure. All we need to do is vasodilate. So if resistance and vasodilation would decrease resistance, right? Blood can get through a dilated vessel easier than a constricted vessel. So vasoconstriction decreases flow. Vasodilation increases flow. Vasodilation causes a decrease in resistance. Vasoconstriction causes an increase in resistance. So I know this is hard. You have to try to picture it. If you can do that, I think you can get it. So let's go to the next hard topic. Resistance is proportional to blood vessel length. When a vessel is longer, it's harder to push blood through. That's simple, right? And it's a linear relationship. If the distance is twice as long, then the resistance will be twice as much. That's it. And the flow will drop by a factor of 2. It will go in half. So you might imagine a giraffe has a lot of resistance with its blood vessels. Turns out giraffes have really wide diameter blood vessels. They also have exceedingly high blood pressure. What about this relationship, resistance is proportional to 1 over r to the 4th? That's pretty tough. That's down here on the bottom. Resistance is proportional to 1 over radius to the 4th power. What that means is, qualitatively, it means that a small change in blood vessel radius will give a big change in resistance. I'm going to say it again. A small change in radius gives a big change in resistance. What this means is a little bit of vasoconstriction will really drop flow a lot. A little bit of vasodilation will really increase flow a lot. I gave you an example in lab, and I'd really like you to reread that and think about these changes. Now, I tried to also give you some new material in this review. And what you can see here is that I made 2 graphs, and I just used Excel, really simple. So look at this graph on the left. As the radius gets bigger, I go from 1 to 10. Look at this blue line. The flow goes from about 100 all the way up to over 100,000. Tremendous. The red line is if the flow was only linearly proportional to the radius. So in this case, the line looks flat, but it really isn't. Flow goes from about 100 to about 1,000. So 100 to 1,000 or 100 to 120,000. Which do you prefer? Well, through evolution, we prefer 1 over r to the 4th. So flow decreases when radius decreases. We'd have to go this way, right? And flow increases when radius increases. I made 2 graphs, but it really, they say the same thing. I should have been thinking more when I made these graphs. And the linear relationship, it's just flat. Yes, there's an increase, but it's not so dominant. This next slide shows it again. So again, let's just look at the top 2 panels. Flow decreases when radius decreases. So if radius goes from 10 to 1, we go from about 100,000 down to about 100. Let's look over here, and if it's linear, almost nothing happens. I drew this graph again because the scale is so different. So radius is decreasing and flow is increasing. So notice we go just from 1,000 to 2,000 when we go from 10 to about 5. Big difference between linear and 1 over r to the 4th. So take home message. Blood flow decreases when vessel radius decreases. A different way to say that is that vasoconstriction decreases blood flow. Conversely, vasodilation, so the tube gets bigger, causes an increase in flow. And this is important. The relationship is to the 4th power, meaning 1 over r to the 4th. You don't have to be a mathematician to know that small changes in radius, a small vasodilation, will give us a big increase in flow. That's the message for most of us, if we're not a cardiologist. Okay. More objectives. Know the approximate blood pressure for major arteries that perfuse organs, systemic circuits, and pulmonary circuits. Now I know there was a lot to do in lab, and everybody didn't do this. I also know that this is an objective. So it's my job to test you on this. Look at figure 21-10 in the textbook. you'll see that the aorta has a pressure, an average pressure, right? That's systolic and diastolic. We can create this average of roughly 100 millimeters of mercury. Now, capillaries are closer to 30 millimeters of mercury, and the vena cava is really close to 10 millimeters of mercury. So there you go. The pressure going from the aorta to the vena cava is roughly 90 millimeters of mercury. The pressure to get through the capillary beds, it goes down about 10. Now, muscular arteries and arterioles. Almost all the blood vessels that you learned are muscular arteries. That means if I ask you the blood pressure, then you, by all rights, should tell me it's 70, 80 millimeters of mercury, something like that. If it's a big one, subclavian, maybe 90. You better know that the subclavian is going to be a greater pressure than, say, the axillary or the brachial, and certainly more than the radial and the ulnar arteries, right? The radial and ulnar arteries would be down here, whereas the aorta is up here. Okay, I hope that helps. I think that's all I have. Good luck. This took longer than I expected. Bye now.